Greetings, everybody. My name is Stefano da Sacco, and I'm an assistant professor at Children's Hospital Los Angeles. First of all, I would like to thank the organizer for giving me this opportunity to present our work. And today I'm going to talk about the Glomerulus-On-A-Chip as a platform for disease modeling, drug screening, biomarker discovery, and mechanistic studies. These are my disclosures. And before starting, let me just tell you a little bit about the kidney. In the kidney, the nephrons are the filtration unit of the kidney, and they are composed by glomerulus and tubules. In adults, there are no new nephron formation, and that means that loss of nephron function leads to development of chronic kidney disease, a condition that affects about 14% of Americans. Within the glomerulus, there is the glomerular filtration barrier that is composed by podocytes, fenestrated glomerular endothelial cells that are located at the opposite side of the glomerular basal membrane. All these three components are extremely important and essential for the function of the kidney. And damage to one of them leads to injury and defects in all the others. Why is recreating a glomerulus so difficult? There are three main reasons. The first one is an architectural challenge. These three components that we were saying are closely in contact, and this allows interaction and continuous crosstalk. The second reason is the source of the cells. It has been a very problematic issue in many fields, and in the kidney it makes no exception. And this is particularly true for podocytes. And the third reason is that most of the studies and the knowledge that we have about the kidney comes from studies in mice. And as we have learned and we keep learning, mice are different from humans. And what we might understand in mice might be useful, but might not be sufficient for our understanding of human physiology. To solve the first issue, we tried different approaches. We started in the beginning with 2D approaches in a plate, or maybe 3D systems like hanging drops, but they didn't work very well. Luckily, very recently, there's been the huge development of systems that are known as microfluidic systems that are the basis for the development of organ-on-a-chips. And we decided to use a commercially available organ-on-a-chip that is made by 3 channels, as you can see in the central panel. Channels that in our system we call C, E, and F. And I will explain the reason about that later on in the presentation. This system allows control of channel filling, taking advantage of surface tension and the formation of a meniscus by collagen that can be loaded in channel E. That is E for extracellular matrix. In our system, we decided to try an approach that would allow us to seed the cells without the presence of any membrane in between the two cell types. In fact, there are different Glomerulus-On-A-Chip systems that have been developed by our colleagues, but most of them present the two cells that are seated on the opposite side of a porous membrane. As I was mentioning before, podocytes and glomerular endothelial cells are closely in contact, and we were trying to mimic this specific. And as you can see here, we use the collagen as just a backbone for support of the seeding of podocytes, a single layer of podocytes, on top of which we seed the glomerular endothelial cells. This allows us to don't have any membrane in between the two cell layers. The second issue was the sourcing of the cells. In our work, we decided to try different types of podocytes. We started with human amniotic fluid-derived podocytes because during development of the fetus, the amniotic fluid contains a lot of cells from different organs that we have shown before. We were able to isolate kidney progenitors based on the selection for CD24, ubiquitin, and podocalexin. And we were able to show that we are able to differentiate these cells and acquire a morphology that is compatible with the one of podocytes, expression of podocyte marker like nephrin, podocin, and WT1, and also, more importantly, collagen 4 production. Collagen 4, alpha 3, 4, and 5 are the main constituents of the glomerular basal membrane, and they are produced only by podocytes in the kidney. We also confirmed that these cells were really podocytes by functional studies, including response to toxicity, response to angiotensin, and cell cycle analysis. We also wanted to use true podocytes, and so we developed the system to isolate primary podocytes and glomerular endothelial cells from kidneys that are discarded for transplantation. These kidneys that are not presenting any damage that is peculiar of kidney disease were digested to obtain glomeruli, and from the glomeruli we isolated CD31 cells that identify glomerular endothelial cells and nephrin-positive cells that identify podocytes. We have performed a very wide analysis of these cells to confirm their phenotype. We also use the golden standard of podocytes that are immortalized in human podocytes. Those are the podocytes that have been used for many years for studying podocytes in vitro. So the first thing that we did was we decided to seed only podocytes and look at how well they were performing in our Glomerulus-On-A-Chip. And as you can see here, all three lines were able to maintain expression of nephrin, confirming that these podocytes were maintaining their phenotype and they were healthy. We also seeded only human glomerular endothelial cells using a specific glomerular endothelial cell media, and we let them grow confluent and form a capillary-like structure, as you can see from the panel B in our figure. You can see that the red fluorescent cells are able to form a capillary that you see in channel C. Channel C is called C just for capillary, and you confirm that the cells maintain expression of CD31 and respond to staining for with germ agglutinin that identifies the glycocalyx. So once we have proven that these two cells were able to be cultured alone, we decided to put them together. And so we seeded first the podocytes, and after 24 hours, we added the glomerular endothelial cells. As you can see from the pictures here, these two cells form two layers that are contiguous and are in contact, suggesting that they are able to interact very well. We have shown that this is particularly true for the human amniotic fluid podocytes and for the primary podocytes. Immortalized podocytes, I have to say, responded a little bit less well to our system. We also wanted to confirm that together with the two cellular layers of the glomerular filtration barrier, we were able to find the glomerular basement membrane, that is the third important component of the glomerular filtration barrier. As you can see here, we have confirmed the expression of collagen 4 alpha 4 and collagen 4 alpha 5, as well as laminin alpha 5. And what is important is that this one is a de novo production, because there is no collagen 4 of any kind in our chip before the seeding. So we have shown that our chips are able to maintain the appearance of being glomerular filtration barrier-like, but are they really working like a glomerular filtration barrier? And one of the most important roles of the glomerulus is to prevent protein loss from the blood. And in fact, proteinuria is normally used in the clinic to assess renal damage. And so our question was if our system was able to retain protein on channel C, that is the capillary channel. As you can see here, what we did was we added Fit-C albumin, that is to the perfusion channel, that is channel C, at the physiological concentration of 40 milligram per ml, that is the concentration that we find in the human blood. As you can see in the first three panels, B, C, and D, the albumin is retained on the channel C without flowing through the channel E and the channel F. Channel F is called like that because it's what we use to collect the filtrate. That's why channel F. And so the reason for the maintenance of the albumin in channel C was definitely the presence of the cells, because when we added albumin to chip with collagen but no cells, the albumin was diffusing immediately into the other channels. The same happened when we were substituting glomerular endothelial cells with human lung endothelial cells, or when we were substituting podocytes with the human fibroblasts, suggesting that not only the presence of cells, but the presence of the right cells is essential for the function of the glomerular filtration barrier in vitro as we designed it. As you can see here, we also performed the statistical analysis, and we confirmed that the 3 podocytes line with the glomerular endothelial cells were outperforming all the other conditions. We also performed long-term selectivity assessments over 28 days, and we performed this albumin test every week. And as you can see here, primary podocytes and human amniotic fluid podocytes generated Glomerulus-On-A-Chip systems were able to maintain an albumin perm selectivity over time, while human immortalized podocytes were losing very quickly their ability to retain albumin. We also checked apoptosis at 28 days, and this one is just a representative picture, but we performed it for all the 3 lines. We didn't see really apoptosis after 28 days, suggesting that our cells are still viable for a long time. So that being done, we decided to look at some response to injury. And the first system that we used was a nephrotoxic injury caused by puromycin aminonucleoside, or PAN. In this, puromycin aminonucleoside is a very well-known nephrotoxic agent that is able to damage the podocytes, disarranging the podocyte cytoskeleton, leading to disruption of the slit diaphragm. The slit diaphragm that is an essential component of the podocytes layer. As you can see here, after addition of the puromycin aminonucleoside, we have a disruption of the cytoskeleton, as you can see in the panel on the right, with formation of those classic rings that define damage in the podocytes. When we did that in the chip, we were indeed able to cause a response in all the 3 cell lines, and with loss of albumin that is shown in panel K and Q after 60 minutes. And we also showed that there was a disruption of the cytoskeleton by staining for F-actin. We were very intrigued by our response, by the response of our system to injury, and we decided to see if we were able to use this system to model different kidney diseases. The first one that we chose was membranous nephropathy. In membranous nephropathy, that is an autoimmune disease, the body develops antibodies against proteins that are normally expressed by podocytes. Mostly, the target proteins are PLA2R or thrombospondin 7A, even though there are many other proteins that are being discovered lately. This binding of antibodies against proteins leads to the deposition of immunocomplex that leads to podocyte injury, proteinuria, and ultimately renal failure. The first thing that we wanted to confirm was that our podocyte lines were expressing PLA2R, and indeed, all the 3 lines were expressing PLA2R, that is the target that we have chosen. However, please notice that the immortalized podocytes that are in the center were expressing a lower amount of PLA2R, and this becomes important when we will look at the results afterwards. We also confirmed that by following the addition of 0.5% human serum from patients suffering from membranous nephropathy within the chip, we were able to show that there was both deposition of total and IgG4, total IgGs and IgGs4, suggesting that indeed the antibodies present in the human sera were able to target the podocytes and bind them. We also confirmed that this was leading to an increase in loss of nephrin. That is an essential protein that is paramount for the function of the podocytes. When we went and looked at the functional effect of PLA2R binding on the chip, we found that compared to chips where the sera was coming from healthy controls, the sera from membranous nephropathy patients was able to trigger a response that was leading to albumin loss in the chip. This one was true particularly for the amniotic fluid podocytes and the primary podocytes, while the immortalized podocytes were not responding as well. This could be, though, possibly because of the lack of high expression of PLA2R in this cell line. We were very excited to see that we were able to find a response of the chip from to membranous nephropathy sera. But we were even more excited when we went back and looked at the correlation between the proteinuria that we were measuring on the chip and the proteinuria on the patients at the time of the blood drawing. As you can see here, both for the kidney podocytes and the primary podocytes, we were able to find a clear correlation, a high correlation between the measurement on the chip and the measurement in the proteinuria of the patient, suggesting that we can use this system not only to model disease, membranous nephropathy as a disease, but also as a potential diagnostic tool that replicates very well what is happening in vivo. We also wanted to confirm that our system was specific, and so we chose to expose our chips to the sera of different kidney diseases, including FSGS. These samples were from patients in remission, so they were not known to have, they were not expected to have any molecule or anything that would trigger a response. We also tested the sera from Alport patients. In this case, Alport patients is a genetic disease. We will talk more about that later, but it is not known to contain anything in the blood that would cause podocyte damage. And we also looked at sera from patients from polycystic kidney disease. And as you can see from our results, none of those sera was able to trigger a response and loss of albumin, suggesting that our system is very specific and responds to specific damages that are and not just to human sera per se. We also wanted to confirm that our system was potentially useful for drug testing, in particular in the preclinical environment. And so we decided to test the alpha MSH, that is a ASTH analog that is being used for the treatment of membranous nephropathy when corticosteroids are not efficient. And in our case, the alpha melanocortin 1 hormone was able to lower and prevent the loss of albumin in our chips when it was administered at the same time with the MS patient sera, suggesting that indeed we can use our systems also to study the response to drugs and how efficiently this drug could rescue patients before moving to clinical trials. As I mentioned before, we are going to go a little bit more in detail about Alport syndrome. Alport syndrome is a genetic disease that is caused by defects in the production of collagen 4, in particular collagen 4, alpha 3, 4, or 5. During normal development, in our kidneys, we start with the presence of collagen alpha 1 and 2 in the glomerulus and in the glomerular filtration barrier. But over time, this collagen 4 alpha 1 and 2 is substituted by collagen 4 alpha 3, 4, and 5 that becomes predominant during adultness. However, in Alport syndrome, where there is this genetic disease, what is happening is that the body is unable to make the switch and substitute the collagen 4 alpha 1 and 2. This leads to defective glomerular basal membrane, as I was mentioning before. When there is one defect to one of the components of the glomerular basal membrane and the glomerular filtration barrier, all of the other components are affected. And indeed, the podocytes and glomerular endothelial cells in Alport syndrome are affected by these defects in the glomerular basal membrane. And this leads to a split thickened basket weave glomerular basal membrane, as you can see in the picture on the right by scanning electron microscopy. So, to model this one, we were lucky enough that we were able to obtain amniotic fluid from a fetus that was carrying a mutation for collagen 4 alpha 4. Using the same approach that we used to isolate the human amniotic fluid kidney progenitor, we isolated amniotic fluid progenitors from these Alport patients by co-expression of CD24, obicadarin, and podocalexin. There are about less than 1% of the cells are expressing these markers, and we were able to expand them before differentiation. Once we used those defective podocytes on the chip, we were able to show that these podocytes that were carrying this mutation for collagen 4 were leading to the production of the defective GBM, leading to both proteinuria on the chip, as you can see on the panel on the right. And this one was a statistically significant difference. We also decided to look at diabetic nephropathy, that is very common damage for the kidneys, in particular due to the high incidence of diabetes in our populations. And as you can see here, when we had a sufficiently high glucose amount in the media, and this one was 20 millimolar, we were able to show that after 72 hours, we were able to trigger a damage that was leading to loss of albumin. In conclusion, I would like to say that we have successfully established a barrier-free system that is formed by human podocytes and human glomerular endothelial cells, and we do that by using 3 different human podocytes lines. The glomerular endothelial cells and the podocyte-like cells tend to remain compartmentalized and interact while maintaining their specific identities. We have also found evidence of the position of a glomerular basement membrane-like structure in our system based on the production of collagen 4, alpha 3, 4, and 5, and laminin alpha 5. More importantly, we have shown that the newly formed glomerular filtration barrier structure is impermeable to albumin and is susceptible to injury. This one leads to a damage to the filtration barrier that again leads to increased permeability to albumin. And this one suggests that our system is feasible for toxicity studies. Our system, as we have shown in our model of membranous nephropathy, is patient-specific, and the results highly correlate with clinical data. Our system is also versatile. We have shown that we can model different diseases, including diabetic nephropathy, various chronic kidney disease, and it can be used for disease modeling. And our system is also feasible for as a platform for drug testing. In conclusion, I would like to thank everybody that has contributed to this work. That could not be done without the support of our chief, Roger De Filippo, and my colleague, Laura Perin, that had a pivotal role in the development of this system, and all the people that have worked in this project, including Dr. Zhang, Dr. Pletorzian, and Dr. Villani. I cannot forget our collaborators, both at USC and around the US and in Europe. And in conclusion, I would like to thank the GOFARR Kidney Fund and the NIH for supporting our research. Thank you so much, and I'm happy to take any question.